In G-protein linked receptors, you will have the G-protein coupled receptor.
This receptor is located on the plasma membrane surrounded by the cytoplasm and extracellular fluid.
Then it's followed by a G-protein that has a GDP molecule attached to it, making the protein inactive.
And then an enzyme is present within this signal.
Now, a signaling molecule would attach itself to a G-protein receptor.
This causes the receptor to change its shape and causing the G-protein to attach to the receptor.
This causes the receptor to become activated, making the GDP molecule change to GTP, activating the G-protein.
Now that the G-protein is activated, it will bind into the enzyme, triggering the enzyme to change its shape.
The enzyme is now activated, triggering a cellular response.
GTP is then hydrolyzed back into GDP from the G-protein, causing the signaling molecule to detach from the receptor, releasing a phosphate.
Then the G-protein and receptor are turned back into inactive.
This changes the enzyme, and the G-protein leaves the enzyme, causing the pathway to recycle.
The next pathway is receptor tyrosine kinases.
Tyrosine kinases occurs in the plasma membrane, and it has two tyrosine kinases receptors that are binded to it.
These receptors each have 3 smaller tyrosines attached to them.
Then in the bottom half is the cytoplasm. In the cytoplasm, we have 2 inactive proteins.
And on the top, we have the extracellular matrix. In the extracellular matrix, we have 2 ligands present, which is our signaling molecule.
To get started, we have 2 ligands that move to the tyrosine kinases receptors, and it attaches together.
These receptors are now activated, causing a change, making the receptors bind together, causing a cellular response.
Then ATP will start to come over into the reaction, and it starts to phosphorylate the tyrosines on both of the receptors.
With this reaction, it leaves off 6 phosphates and a ADP.
This then triggers the inactive proteins to come over and to attach to the phosphorylated tyrosine, activating a cellular response.
The ligands then remove itself from the receptors, resetting the pathway,
showing that this pathway is recycled and can be repeated.
Our last signaling pathway is ligand-gated ion channel.
Ligand-gated ion channel occurs in the plasma membrane, and within the plasma membrane, we have a ligand-gated ion channel receptor that is closed.
Then in the extracellular fluid, we have a signaling molecule ligand and our ions present.
The pathway starts off as a closed receptor. Then the ligand, our signaling molecule, will bind to the receptor.
This causes the channel receptor to open up, allowing specific ions to flow through the channel.
This causes a rapid change in the concentration of the ion.
This change causes a cellular response affecting the activity of the cell.
The ligand will eventually remove itself from the receptor.
This causes the channel to close back up, stopping the ions from entering the cell, basically resetting the pathway, showing us that the pathway can be repeated.